everyone hear me, including individuals at the back? Okay, unfortunately today Dr. Roper could not be here. She is currently at the HLB meeting at the Riverside Convention Center, but she should return for the last lecture of the quarter on Friday. A couple announcements. I know all of you are well aware that your final is a week from yesterday. The exam is not cumulative. It is the same format as all the other exams. Scantron will be provided. Also, remember to bring a number 2 pencil. And if you don't have a number 2 pencil, please raise your hand and one will be provided for you. We should have a number of them. Last exam, a number of students used pen. This is a pencil. So the Scantron machine sounded very unique, to say the least. And we had to go back and grade the entire exam by hand. So to prevent that, please remember to use a pencil. Remember, a review session will be held this Friday from 6 to 7:30, I believe in Borbach. The number escapes my mind right now, but you can probably check the announcements or email your TAs and they'll provide you more information. So, without further ado, today we're going to talk about the first part about microbial ecology. Alright. Now, in ecology, microbes play a very important part. A larger part than more than you would actually think, unless you are a microbiologist. They shape the environment of what the plants and animals, the other larger organisms, interact with. Because microbes recycle organic matter, organic material, and we'll go into more detail about that in today's lecture as well as Friday's lecture. And they do certain things in the environment that you don't think of at all. Like, for example, they can shape crustal rock. So, the theme that I know you've been hearing from a lot from this quarter is that microbes are everywhere. They're found in almost every single environment on the planet, and they fill almost every single niche. They do something in that environment to survive. They have some kind of function. Now, so basically, any energy acquired by these by the microbes, as well as the elements they assimilate, they turn it into biomass, and that biomass circulates throughout the ecosystem and is used by other microbes or other larger organisms. Now, what determines what microbe can do what in the environment? That's in its genome. Remember from just basic biology and from the beginning of this class. The genome, the genetic material of that microorganisms determines what type of enzymes that microbe can produce, and those enzymes produced by that microbe allows the microbe to interact, thrive, and survive in that environment. Now, when it comes to interactions in the environment, what types of interactions are there in terms of between microbes with each other or with the external environment? Well, there are a couple of terms you need to know. I highly recommend you know these terms for a certain exam coming in about 6 days. But basically, some of the terminology is one: assimilation, the process by which organisms acquire an element to build into the cell. Now, this is often used to describe carbon and nitrogen fixing organisms, and they use that to produce biomass. These are your primary producers in the environment. For bacteria, these are your algae and your cyanobacteria. And then the next group would be dissemination, the process of breaking down organic nutrients into inorganic minerals, often through the process of oxidation. And then these compounds are used by others in the environment. Now, for the term biomass, Remember, that's considered as of all the bodies of living organisms in that environment. And then to obtain energy, the flow of energy material, i.e. biomass, the interactions of all the organisms and microorganisms in an environment is, they all interact with what is called a food web. And you should be familiar with food webs from the previous courses here, but in case you aren't, or just like a refresher, remember a food web is a description of the interactions and how organisms consume each other and their products. And the level of consumptions, who consumes what, in a food web is determined by its trophic level, which is basically the level of consumption of an organism of each level. Because basically, at a certain level, organism will consume usually the biomass of another organism in a previous level. And then remember, just like from your ecology, I don't know if you've taken the ecology coursework here or not. Remember, only a fraction of the energy is obtained by that organism as it eats its lower trophic level. Most of it's released in the form of CO2 for respiration to provide energy. Now, when it comes to microbes, there was a Dutch microbiologist named Cornelius van Meel, and he was the first scientist to show that bacteria in soil and water can conduct photosynthesis without producing oxygen. And how the bacteria did that is that it used, as an electron donor, it used hydrogen sulfide instead of H2O. And his postulates stated that every component or product of a living cell can be used as a source of energy or carbon by a microorganism found somewhere in the environment, in the biosphere, or in the planet. In other words, if an energy-yielding reaction exists, a microbe has evolved or will evolve to use it. And then that's one of the primary reasons why microbes are found everywhere on the planet. Now back to some basic ecosystem information. So as you all know, all food webs begin with the primary producers. And again, the primary producers, when we talk about microorganisms, these are your cyanobacteria, and the word bacteria that can use photosynthesis, and your algae. And basically, there are two things that they do that's required by everyone else, that they absorb energy from outside the ecosystem. For example, that could be sunlight or phototrophs, and they assimilate materials into the biomass, as you can see here. Now, remember, there are, on top of the primary producers, there are the consumers. The next level up from the primary producer is the grazers. The microbes or individuals or organisms that feed primarily on the primary producers. And then you have the predators. And the predators feed on the grazers. And then at each trophic level, as those organisms die, they all interact with the decomposers. These are often in the environment, in terms of microbiology, these are your fungi. They break down, but they can also be bacteria. But they break down minerals and return them into the environment for other organisms to use. Now we just described the general food web. You've probably seen it a whole bunch of times in all your other classes, right? You have your primary producers, that's where the biomass is formed, i.e. like plants. Then you have the grazers, your herbivores that eat the plants, and then the predators eat the grazers, and when everyone dies, the decomposers take care of their bodies. Now let's take a look at the food web on the right, the production of biomass let's compare that, the terrestrial, i.e. the forest that's on the bottom, and compare it to the ocean. And the reason why we're talking about the ocean, because microorganisms actually play a larger, more significant role. And then there are certain characteristics about the microbes in that environment, which you'll see by the end of the lecture. As you can see, the primary production of biomass in the ocean, or your primary producers, your CO2 fixers, basically in the forest it's the plants, the trees, the grass, whatever. In the ocean, it's the phytoplankton, the bacteria, archaea, and algae. And then where they exist, we'll talk about in a moment. And if you look at that, out of those primary producers, 20% is consumed by the grazers. That's in the forest, right? Whoever eats the plants. And then for 80% of the rest of the biomass, decomposers will break it down, or you can also include fire and that. However, in the ocean, you have protists and vertebrates taking up 60% of the primary producer's biomass, all those bacteria and algae and archaea, while 40% the decomposers are viruses. And they basically will break stuff down and return it to the environment. Well, that's important because, like we just said, different habitats have different producers and consumers. Again, the marine, the producers are CO2 fixation and biomass production are performed by phototrophic bacteria. And protists and viruses consume the phototrophic bacteria. And while terrestrial, we already know plants get eaten by herbivores. Now we're going to talk about the different interactions from a microbial standpoint that you see in microbial ecology. You already know from other courses, the definition of symbiosis is the association of microbes with another species. That another species could be a larger species like a plant or an animal, or it could be another microbe. The four basic types of symbiosis include one, mutualism, or both benefit. And then the opposite of mutualism is parasitism, where one benefits at the expense of another. And then we'll talk a little bit more about synergism and commensalism, but those two terms are a little more gray, obviously, than mutualism or parasitism. So we'll discuss a little bit about that in about when we get to those slides, basically. So first, we're going to talk about now mutualism, where both partners benefit from a specific association. When it comes to microbiology, some of the most obvious examples of mutualism include lichens. We're going to go over what a lichen is. Basically, it's a combination of a fungus growing together with algae and or cyanobacteria, and what's the benefits taken from each microorganism. We're going to talk about how and why rhizobiums interact with leguminous plants. We're going to talk about the paramecium, myotrichia, which is found in the gut of termites, and why you should care about that. And then we're talking about mycorrhizae. For the first one, lichen. Basically, it's when a fungus grows together with some kind of photosynthetic microorganism, usually a cyanobacteria or a green algae. The bacteria uses photosynthesis to produce oxygen and generate organic carbon and fix atmospheric nitrogen. The fungus provides shelter, water, and minerals. And how can this benefit each one? Well, the symbiosis between the fungus and the bacteria allow the lichen to survive. And they're often the primary colonizers in soilless environments. so in environments where you don't think is a lot of resources, or you don't think where something like would grow. Like for example, if you've ever been hiking or whatnot, or hiking, you see like on some rock surfaces some weird green growth, and it's like, how can something even grow there? Well, that's because it's a lichen that's taking care of that. And then if you were to look at the lichen up close, what you see here is all the different microorganisms that compose the lichen. You have your cyanobacteria, and you have your algae. Remember, they use photosynthesis to provide organic carbon, as well as oxygen if needed. And then you have the fungi, which provides the minerals, the water, and the protection. Now we're going to talk about rhizobia-legume interactions. We're going to talk about legume interactions and how important of these plants are. These are the plants are usually peanuts, soybeans, and alfalfa. And then we're going to talk about briefly, from a basic standpoint, the interaction between the rhizobia and the legume plant, and why is this important. Now the actual process, what you'll see on the next slide, is it looks more complicated than it actually is. So when you study for it for the coming exam, just take it a piece at a time. Don't wait till Monday evening to tackle certain things. Now remember, we're talking about ecology, mutualism. So remember, each partner gives something for the benefit of something. So remember that when you study for the exam. The plant provides a carbon source and shelter. The rhizobia fixes atmospheric nitrogen to ammonia. Usually nitrogen, especially to take it up from, basically nitrogen has to be in a certain form for plants to use so they can grow in the environment. So rhizobia fixes atmospheric nitrogen and provides it to the plant, and that gives the nitrogen, gives a major source of nitrogen to the plant. Now this is very important because the overall, what benefits like you and me, well, I'll explain at the end of this slide. But basically, this is the process that you're going to have to know. How does the rhizobia interact with the plants? Usually, there is some kind of interaction, a signal exchange between the two parties. Often that could be flavonoids from the plant. And then the rhizobium, the bacteria, will bind to the root hair of the plant, as you can see on the right. And then the rhizobia enters the cortical cell through basically almost an infection process. So now, once it is inside the plant cell, it becomes what is called a bacteroid. It's a regular shape with no cell wall. And then this bacteroid is enclosed in a plant-derived membrane. And this is important. And this plant-derived membrane is called the symbiosome. And then at this phase, the bacteria supplies the fixed nitrogen, and we're converting atmospheric nitrogen into ammonia, and the plant takes that up to grow. And then the plant has a protein called oligohemoglobin, which sequesters excess oxygen. Now, the key here is not oxygen, excess oxygen. And then you'll see why in the next slide. And the question is, why should we care about all this? Well, for those of you who have had some farming background or talk, have you ever heard of like when they plant certain crops to help refertilize the soil? Well, what they do is they plant certain plants like these. There's the bacteria that interacts with the plants. You have this buildup of nitrogen in the form of ammonia, and when the plant dies, basically that all that nitrogen that was taken up and built up in the plant is now released back into the soil. But instead of being atmospheric nitrogen, that's now ammonia. So basically, that is now used to basically re-fertilize the entire field, and then now you can grow your next crop on that field again. Now this is a close-up of what the bacteroid looks like within the plant cell. Here's the symbioso membrane, right? Just that larger white circle. Inside those white circles, those weird black ovals or ovally shaped objects, those are your bacteroids. Now we're going to come back to remember what I said. The plant, the bacterium supplies the fixed nitrogen, and then the plant has a protein called a ligohemoglobin, which sequesters excess oxygen. And here's another version of that. But then the significance of that is nitrogen, that enzyme that the bacteria uses to produce ammonia from atmospheric nitrogen is called nitrogenase. However, too much nitrogenase is damaged by oxygen. Which you would think is, well, how will we just eliminate all the oxygen and then we don't have to care about that? The problem is the bacterium, the rhizobia, needs oxygen for bacterial respiration. So you can't take away all the oxygen or the bacteria will die and you don't get any ammonia production. So there is a protein, the leg hemoglobin, it contains iron that sequesters excess oxygen. And think of it as the plant version of hemoglobin. Spores to spread. And then a much more, you probably hopefully might have heard this on the news a couple of years ago, the Dutch elm disease, which is another fungus-based disease caused by in plants, which, as you can see here, that tree doesn't look that healthy compared to its neighbors. And what it does is that the interaction of the fungus with the plant causes the tree leaves to become yellow a lot earlier than it normally does through the seasonal process. So it will become more yellow like the middle of summer for some reason. And then the leaves will start dying. Right? Eventually the fungus will spread throughout the entire tree, so you'll have basically almost all the leaves dying and coming off the tree. That's why this gives it this dieback. Eventually it will spread to the roots and the entire tree will die. This is caused by an Ascomycetes microfungi. Now when we talk about parasitism, usually you think, when you talk about it, at least from a human's perspective, that's all gloom and doom, right? Something's dying, something is changing, we can't use it. Well, remember the earlier part, I said 5 minutes ago, parasitism in plant pathology is primarily fungi, but that doesn't mean bacteria and viruses can't be a part of that group as well. And what you have here is the viral infection of tulips. Now, why do you usually care about this? A tulip in its natural healthy tulip will never be one color. Will only be one color. However, when a virus, a certain virus, it's called the tulip breaking virus. Gee, that's an original name. What it does is that once it infects the tulip, it causes basically an uneven pigment accumulation or a lack of in the epidermal layer, for lack of better terminology, of the flower portion. The flower portion of the tulip plant. And now, even though the tulip might not be worth, it's different from normal, but however, depending on how the virus accumulates the pigments in only certain parts of the flower plant, actually the multicolored tulips are worth a lot of money. So, especially in Europe. So that's a way how certain people have used this process of parasitism and plant pathology to make a good, make a quick buck. Now we're talking about synergism. And basically, when it comes to synergism, is that usually you have two species, right? Now they can live separately from one another. They don't need to interact, and they can still be happy and live about their lives. However, when they interact, they usually there's an increased effect that's usually beneficial to both due to their cooperation. The example given here is your bacteria in your gut. One of their byproducts is used by colonic methanogens in your bacteria in your colon, and this basically creates a proper environment for both. So basically, the product of the human colonic bacteria, a byproduct of the first group, can be used by the colonic methanogens. Commensalism is a little more tricky because usually an interaction is only when one side benefits and the other one doesn't care, or it's not harmed. Now this can be hard to study because the argument is if one benefits but neither are harmed, why should the group that isn't being benefit but not harmed care, right? So it's always like hard to define when something is shown as a commensal relationship. It's hard to define, is it truly a commensal relationship or not? Yes or no? The example given is like the Bacteroides. This is a natural flora of your gut. It was once considered commensal. It's just there in your gut. We don't care. It just hangs out in your gut. It lives off the byproducts that you digest in your food, but it doesn't give you any benefits, so why should we care about it? However, when we talk about recent research when it comes to microbiomes, particularly your gut microbiome, has shown that actually Bacteroides aids in metabolism and in digestion, in keeping a health and with the proper intake of nutrients in your gut. And then when you travel and you go through jet lag, that actually disrupts how well the bacteria can break down your food. So now, since we talked about all the different types of interactions, mutualism, parasitism, commensalism, we're going to turn into marine microbiology. Because as you saw earlier, there's a little microbes play a little bit more of a role, obvious role, or a clearer role versus microbes in another environment. Before we do that, we got to know about the basic environment of the ocean when it comes in terms to microbes. Now we talked about the water column, right? That's in the ocean. It's divided as you increase in depth, as you go straight down. Basically, it's divided into distinct regions, and there's a reason for that. The nearest region up top in Houston, this is basically 10 microns, that tiny amount right at the surface of the water. This is basically the air-water surface, air-water interface. This contains the highest amount of concentrated microbes in ocean water. This is contained where most of your algae and the protists are. You go a little bit more down, it's the euphotic zone. This receives light, so allows phototrophs to grow. And then depending on how close you are to the coastline, this can change because you have a buildup of sediment that might affect the amount of light. Below that, you have the aphotic zone. This is below where light can reach. So only heterotrophs, right? Consumers of basically microbes that can use organic material, and lithotrophs can grow. Remember, you can't have your phototrophs here because there is no life. And then you have your benthos, the bottom of the ocean. Ocean floor plus the sediment below the surface. And this is going to be very important, we'll discuss in a moment. And around here, you have certain communities, and then that microbes will interact. Usually you find any type of living thing at the bottom of the ocean, it's surviving down there due to the help of certain microorganisms. And we'll go over that, a couple examples of that in a second. And then here is just an example of what you have of the water column. And you would want to know the level, what's happening at each level, as well as how much the level extends to. Now, why should we care about, as a microbiologist, this is a microbiology class, why should we care about the ocean? Well, a lot of many unique microbes that have unique capabilities actually survive are found in the ocean. At the bottom, where we talked about the benthos, in that layer, that's where you have your birophiles. Remember, a birophile is a bacteria that can survive at extraordinarily high pressure. At the seafloor, any microbes also support the psychrophiles. These are the bacteria that can survive in low temperature. They thrive in it. Most bacteria or microbes will die. Then in the hydrothermal vents, you have your thermophiles. Any bacteria found there can withstand extraordinarily high temperature. And that's where you see it being studied there. And then, as you can see, how do these microbes that exist as part of the environment, in forms of mutualism or commensalism, those bents and cold seeps, you have a lot of sulfur oxidizing bacteria, methanogens and methanotropes. Basically, when it comes like an example against the tube worm, they oxidize the hydrosulfide and the methane and provide resources for the tube worms to actually survive. An example of that is, as you can see here, this is found near the deep sea hydrothermal vent. Because the argument was when this discovery was made, was okay, you have at this area, it's super deep, so it's high pressure, it's really hot. Why do you have this giant thing of worms hanging around near this supposed to be really nasty environment? Well, we found out that due to the help of bacteria, these worms can actually gain nutrients from that really harsh environment and survive, as you can see here. Now, the study of it can be much more difficult than it actually is. Usually, humans obviously can't go down there. So we're going to usually, you need special equipment. This is Alvin, the submersible. For those of you, probably, I'm guessing almost everyone here was born after 1987. But basically, that submarine was used to help find the Titanic. And basically, and it's also used to do research at the bottom of the ocean or areas of very high pressure. And that little bottom there, unfortunately, Dr. Roper isn't here to explain the story. Dr. Roper's friend was a researcher. I'm not sure if the friend was a researcher or maybe like an undergraduate researcher in that group. And basically made that cup for Caroline, so for Dr. Roper. As you can see, her friend put her name on it and decorated it nice and neat, and then took it to the bottom of the ocean. And as you can see, it got squished and that's a dime right next to it. And that used to be a standard styrofoam cup that you get from the restaurants. And as you can see, by the amount of pressure, it squished the cup. So then here it is again. And here it is, again, to sample bacteria at that depth. Here's a special bacterial sampling device. It's probably much more complicated, what we'll be having you do for those of you that decide to take the lab. And how does this come all about? Well, as you recall from the beginning of class, many microorganisms are currently unculturable. Now, the biggest discrepancy, greatest visible discrepancy where this is seen is in the ocean. When you try to isolate microorganisms from the ocean water, if you were to take basically just take a pipette, take a small amount of water straight out of the ocean, put it under a microscope, you see all this stuff floating around. However, then you put it on a plate, and guess what? You see nothing. So how does that explain the two? You know something is there, but you can't culture it in vitro on a plate in the lab. So how can you study then that environment? What came about was then the idea of metagenomics. Instead of trying to grow up a microbe on a plate, you see a colony. Basically, thanks to the evolution of technology, especially in molecular biology, we're just, instead of showing that some kind of microbe is there by having it grow on a plate, we're just going to sequence all the DNA sequences in the community. And once we find all the DNA sequences, that will tell us what microbes are there. And then from there, you can expand your research, your needed research, without needing an actual physical specimen on the plate, by like transcriptomics, finding out what genes are turned on in the environment just by sequencing everything. But we'll talk more about that later. And then what we see here, based as when they started doing that research, especially with marine communities, is that usually the genes that are the most highly expressed are the ones rarest in the genome. And because of that, you could say, well, what's so special about this one particular gene? Because it's found at very little, it's found number of microbes that have it is very minimal, but the number of transcripts, its expression is super high. That means that gene must do something important. And if you want to know that, you have to come to class Friday. Are there any other questions? Concerns?